Cellular respiration. Aerobic respiration is common to nearly all eukaryotic organisms. It can be divided into three parts. 1. Glycolysis. 2. Krebs cycle. 3. Electron transport phosphorylation. What is glycolysis? Glycose means sugar. Lysis means splitting. Glycolysis involves a series of reactions carried out by a group of soluble enzymes located in both the cytosol and the plastic. Glycolysis occurred in cytoplasm. Input is sucrose. Outputs are pyruvate, ATP, and NADH. There are two types of phases: initial phase and energy conserving phase. Initial phase of glycolysis. Substrate from different sources are channeled into triose phosphate for each molecule of sucrose that is metabolized. 4 molecules of triose phosphate are formed. The process requires an input of up to 4 ATP. In the early steps of glycolysis, sucrose is broken down into the two monosaccharides, glucose and fructose, which can readily enter the glycolytic pathway. Sucrose synthase localized in the cytosol is used to degrade sucrose by combining sucrose with UDP to produce fructose and UDP glucose. Glucose and fructose are then phosphorylated in a reaction that uses ATP. UDP glucose converted into glucose 6-phosphate by UDP glucose pyrophosphorylase. Glucose 6-phosphate can be converted into fructose 6-phosphate. Plastids convert starch into the glucose 6-phosphate using a separate set of glycolytic isozymes and release to cytosol. Fructose 6-phosphate convert to fructose 1,6 biphosphate. Fructose 1,6 biphosphate then split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Between these two compounds, reversible reaction can be happen. In here, triose phosphate isomerase enzyme is in energy conserving phase. In this phase, triose phosphate is converted to pyruvate. NAD plus is reduced to NADH by glyceraldehyde 3-phosphate dehydrogenase. ATP is synthesized in the reactions catalyzed by phosphoglycerate kinase and pyruvate kinase. Once glyceraldehyde 3-phosphate is formed, the glycolytic pathway can begin to extract usable energy. The enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes the oxidation of the aldehyde to a carbolic acid, reducing NAD plus to NADH. Of glycolysis catalyzed by phosphoglycerate kinase, yielding ATP and 3-phosphoglycerate. In each sucrose molecule, 4 ATP are generated. This type of ATP synthesis, called substrate level phosphorylation, involves the direct transfer of a phosphate group from a substrate molecule to ADP to form ATP. In the following reaction, the phosphate on 3-phosphoglycerate is transferred to carbon to and a molecule of water is removed, yielding the compound phosphophenol pyruvate. As substrate, the enzyme pyruvate kinase catalyzes a second substrate level phosphorylation to yield ATP and pyruvate. At the end of the glycolytic sequences, plants have alternative pathways for metabolizing PEP. PEP is converted to oxaloacetic acid. OAA is then reduced to malate. Malate then enters to mitochondria to use in citric acid cycle. In the absence of O2, fermentation can be occurred. By alcoholic fermentation, the two enzymes pyruvate decarboxylase and alcohol dehydrogenase act on pyruvate, ultimately producing ethanol and CO2 and oxidizing NADH in the process. On to the final production. ATP production for sucrose in glycolysis. 4 ATP are produced by 4 substrate level phosphorylation. 6 ATP are produced by 4 NADH. Thank you. Thanks for watching. We are from Group C2.
Created using Powtoon.